Predicting the structure of large protein complexes using AlphaFold and Monte Carlo tree. Search Patrick Bryant, Gabriele Pozzati, Wensi Aditi Shenoy, Petras Kundrotas, and Arne Elofsson. Nature Communications Volume 13, Article Number 6028, 2022. Cite this article. Abstract. AlphaFold can predict the structure of single and multiple chain proteins with very high accuracy. However, the accuracy decreases with the number of chains, and the available GPU memory limits the size of protein complexes which can be predicted. Here we show that one can predict the structure of large complexes starting from predictions of subcomponents. We assemble 91 out of 175 complexes with 10 to 30 chains from predicted subcomponents using Monte Carlo tree search with a median TM score of 0.51. There are 30 highly accurate complexes, TM score greater than or equal to 0.8, 33% of complete assemblies. We create a scoring function, MP-doc, that can distinguish if assemblies are complete and predict their accuracy. We find that complexes containing symmetry are accurately assembled, while asymmetrical complexes remain challenging. The method is freely available and accessible as a Colab notebook, https://collab.research.google.com/github/patrickbryant1/mo/lpc/master/mo/lpc.ipin. Similar content being viewed by others. Accurate determination of protein, ligand standard binding free energies from molecular dynamics simulations. Article March 11, 2022. Machine learning overcomes human bias in the discovery of self-assembling peptides. Article October 31, 2022. Energy refinement and analysis of structures in the QM9 database via a highly accurate quantum chemical method. Article Open Access. July 3, 2019. Introduction. Large protein complexes govern many cellular processes, performing complicated tasks such as mRNA splicing 1, protein degradation 2, or assisting protein folding 3. By incorporating protein interaction information from many co-purification experiments, the Human Protein Complex Map, HUMAP 2.04, provides a set of 4,779 complexes with more than 2 chains. However, only 83 of these complexes are present in PDB. There are only 372 structurally resolved human protein complexes with over 2 chains, and of the 3,130 eukaryotic core complexes in Quorum 5, only 800 have homologous structures covering all chains in PDB, suggesting a gap in our structural knowledge of protein complexes. There are only 265 hetero and homomeric non-redundant complexes in the PDB with 10 to 30 chains. Although it is unknown how many large complexes may exist, following the relationship between the known human complexes from HUMAP and the structural coverage of these, one can extrapolate that there may indeed be a low structural coverage across different species. There are at least 3 approaches 6 for modeling the structure of protein complexes: template-based modeling 7, shape complementarity docking 8, and integrative modeling 9,10. Template-based modeling and docking methods have recently been shown to be outperformed by a combined fold and docking methodology using AlphaFold 11 for dimeric complexes, even if the bound form of each monomer is known 12. Further, few docking programs handle more than 2 protein chains, i.e., these methods are not suitable for building large complexes with no close homology to known complexes. There is currently, to our knowledge, no available docking benchmark for complexes with more than 2 chains, and previous studies only report results on a few examples, 13,14. Assembling large protein complexes with integrative modeling generally requires electron density maps or other experimental information to guide the assembly process 9,15. This type of guided assembly is typically based on a Markov process 9 or Gaussian mixture model 16, where many different potential configurations are explored and scored. This process makes it possible to assemble complexes with up to 1,000 protein chains 17. However, obtaining electron density maps can be very difficult, as some protein complexes are hard to express, purify, and crystallize. Still, many recent assemblies of large protein complexes exist, such as the human nuclear pore complex 18 and 26S proteasome 19. The only deep learning method primarily designed to predict the structure of more than 2 protein chains is AlphaFold multimer 20. This method has been trained on proteins of up to 9 chains, or 1,536 residues, and can predict complexes of up to a few thousand residues, where memory limitations come into play. However, the performance declines rapidly for proteins with over 2 chains, supplementary figure 4. Predicting the structure of larger complexes is thereby currently not feasible. An alternative approach could be to predict the structure of subcomponents of large complexes and then assemble them. We have earlier shown that it is possible to manually assemble large complexes from dimers in a few cases 21. In vivo, all components of large protein complexes do not assemble simultaneously, but stepwise 22, due to the presence of homologous protein chains and potential interfaces that need to be buried before subsequent chains can be added. Here, we explore the limitations of AlphaFold for predicting protein complexes with 10 to 30 chains and create a graph traversal algorithm that excludes overlapping interactions, making it possible to assemble large protein complexes in a stepwise fashion. Results. Here, we begin with an outline of the protein complex assembly using Monte Carlo tree search, MCTS. We then explore the success rates using either AlphaFold multimer version 220 AFM or the FoldDoc protocol 12 using AlphaFold 11 AF. First, we examine the use of predicted subcomponents of the native dimeric or trimeric subcomponents. We then continue without assuming knowledge of the interactions, presenting the final protocol based on all possible trimeric subcomponents. Complex assembly. To analyze the possibility of assembling large protein complexes, we extracted all high-resolution non-redundant complexes from the PDB with more than 9 chains not containing nucleic acids or interactions from different organisms, 175 in total. We start by analyzing the possibility of assembling these protein complexes, assuming that exact pairs of interactions between protein chains are known. Using either AFM or FoldDoc, we predict the structure of all unique pairs of interacting protein chains as subcomponents and create assembly paths from these. As an example, the assembly of 6-ESQ, acetoacetyl coethiolase/HMG coasynthase complex, is shown in Figure 1 using subcomponents predicted with AFM. The process starts from the 2 dimers, AC and CH, creating the trimer ACH through superposition using the chain C present in both dimers. Next, chain L is added through a connection with H, superposition using chain H. After that, chain J through a connection with L. This process then continues until the entire complex is assembled according to the outlined path. Figure 1. Assembly principle for the acetoacetyl coethiolase/HMG coasynthase complex, complex 6-ESQ. Figure 1. The structure of all interacting chains is predicted by protein sequences from each chain and the interaction network. From these predictions, an assembly path is constructed, using the predictions as a guide. In each step, one new chain is added through a network edge, resulting in a sequential construction of the complex. The taken path is outlined in red. The complete assembly is shown in overlap with the native complex, gray. The resulting TM score is 0.93 using subcomponents from AFM, shown, and 0.92 using FoldDoc, not shown. Full-size image. During the first part of this paper, we assume that the interaction graph is known, i.e., we limit the assembly paths only to include interactions existing in the complex. Although this is a simplification, the number of assembly paths is still huge, and it could at least be theoretically possible to obtain this information from other types of experiments 23 or predictions 4. Next, potential assembly paths are created by starting at a randomly selected chain and adding all possible connections through superposition. Often overlaps occur among the predictions due to imperfect subcomponents, resulting in that, for example, atoms from chains B and C occupying the same spatial position in a given complex ABCD. Therefore, an assembly path is discontinued when over half of the alpha carbons from two different chains are within 5 ring of each other. An assembly path is complete when all chains in a complex can be linked together. For 6 ESQ, the assembly results in a model with a TM score of 0.96. Monte Carlo tree search. Due to the high number of possible paths to explore, searching all paths is unfeasible. Therefore, we search for an optimal path using MCTS-24, Figure 2, which has been applied successfully to solve a variety of game-related problems 25-26. Starting from a randomly selected chain node, chains are added randomly to expand the path, thereby creating new nodes. From these expansions, complete assemblies are simulated. Simulations are stopped when no additional subunits can be added. See methods. The simulated assemblies are scored by their cumulative MP-DOC, multiple interface predicted DOC, average interface PLDDT times the logarithm of the number of interface contacts, method section, score, and the scores are backpropagated to yield support for the previous selections. The path with the most support is selected, creating a complex that is the most likely to be correct. Due to the statistical nature of the search procedure, no aspect of a specific complex is being learned in the backpropagation, i.e., all 175 complexes can be used for evaluation. Figure 2. Monte Carlo tree search. Figure 2. Starting from a node, subcomplex, a new node is selected based on the previously backpropagated scores. From this node, a random node is added, expansion. A complete assembly process is then simulated by adding nodes randomly until an entire complex is assembled or a stop caused by too much overlap is reached. The complex is scored, and the score is backpropagated to all previous nodes, which yields support for the previous selections. The final result is that the nodes most likely to result in high-scoring complexes are joined in a path containing all chains. The principle for the complex 6 ESQ is shown. Full-size image. AFM versus AF using pairwise interactions. Only 15 out of 175 complexes could be assembled to completion using native pairwise interactions with both AFM and fold doc. Supplementary Figure 1a. The assemblies based on fold doc and AFM are partly complementary, meaning that the subcomponents of either AFM or FD can be used to assemble a complex. The results suggest that if a complete path can be found, the models from AFM are slightly better, median TM score equals 0.83, compared to the fold doc models, median equals 0.77. The AlphaFold Multimer version 1, AFM, V1, modeling pipeline often caused clashes, supplementary Figure 1b, resulting in atoms from different chains occupying the same positions, which is why a new version was developed, version 2. Defining clashes as atoms from different protein chains being within 1 angstrom from each other, 26.7%, 175, 656 of the AFM dimers contain clashes and 6.3%, 41, 656 for the fold doc pipeline. Even though there are more clashes in the AFM predictions, the final assemblies turn out to be of better quality, suggesting that the subcomponents are accurately predicted. For details, see below. Limited conformational sampling in dimers. During assembly, the additive relative orientation of different protein chains can result in overlaps due to predictions not being entirely correct. One cause of overlaps during the assembly process is due to that not all conformations of chain interactions are correctly predicted, resulting in wrong or missing interfaces in some dimers. As an example, we investigated 1A8R, a homotenmer. When predicting unique pairwise interactions, only one type of dimeric conformations can be found, but in the complex, each chain has at least 2 different types of interactions with other chains. This means that it is impossible to assemble the entire complex from the predicted dimers. The overlapping interfaces can here be circumvented by predicting trimeric interactions, thereby generating alternative interfaces, supplementary Figure 1c. We therefore continue with predicting trimeric subcomponents and use all 3 dimers from a trimer for assembly. Complex assembly using trimeric interactions. Using the fold doc protocol and AFM, all native trimeric interactions were predicted for all complexes. Using the unique native trimers, the clashes are more frequent than using the dimers, 37.1%, 829, 2,234 for AFM, while for the fold dock pipeline, the corresponding fraction is 23.2%, 520, 2,242. All dimeric interactions were extracted from the trimers and assembly paths were constructed as previously. Out of 175 complexes, 58, 33%, and 55, 31%, could be assembled to completion with median TM scores of 0.80 and 0.74 for fold dock and AFM, respectively, Figure 3a, Supplementary Table 1. Compared with the guided dimer TM scores, the guided trimer approach results in 46 additional complexes and a higher median TM score for fold dock, while 3 could not be assembled. For AFM, 43 additional complexes are produced with the guided trimer approach, while 3 are missed, but the median TM score is lower than for dimers. Figure 3. Analysis of assembly success using different methods. Figure 3. A TM scores for the complexes that could be assembled to completion using fold dock, FD, or AFM and predicted native dimeric, native trimeric, and all trimeric subcomponents, respectively. The complete set of complexes from the 3 approaches, n equals 108, is shown, with scores of 0 representing missing complexes for each approach. The points display the TM score of the individual complexes, and the black X marks the average scores. The average TM scores is 0.09 versus 0.10, 0.36 versus 0.33, and 0.47 versus 0.41, using native dimers, trimers and all trimers for fold dock versus AFM, respectively. Fold dock thereby outperforms AFM overall. The median scores are low due to the missing complexes between the approaches. Considering only the successful assemblies using native dimers, trimers and all trimers, the median scores are 0.77, 0.80, and 0.51, respectively. B. Complex scoring using all trimers as subcomponents. Rock a curve, where positives, n equals 30, are complete assemblies of TM score greater than or equal to 0.8 as a function of the average interface PLDDT, the number of interface residues and contacts normalized with the number of chains in each complex, the average interface PLDDT, times the logarithm of the number of interface contacts, and MP doc, CC. The best separators are PLDDT log contacts and MP doc, both with AUC 0.83. CTM score versus the best separator in B, PLDDT log contacts, colored by the fraction of completion for the assemblies, n equals 175. The solid gray line represents a sigmoidal fit, creating the MP doc score, see methods section. When the MP doc tends to be high, so does the TM score and percent completion of the complex. This suggests that MP doc can be used to select when a complex is complete and how accurate it is. DTM score distribution of the complete complexes, n equals 91, assembled using all fold dock trimers and examples at different thresholds. The assembled complexes, colored by chain, are in structural superposition with the native ones, gray. The PDB IDs for the complexes shown and their corresponding symmetries and TM scores are 5TRM octahedral 0.22, 2V5H dihedral 0.45, 7JQZ dihedral 0.51, 5XBB helical 0.82, 2GRE tetrahedral 0.97, and 5T11 cyclic 0.98. At TM score 0.8, the assembled complex is similar to the native one. Full-size image. In many cases, the exact interactions of all protein chains are not known, only that a set of chains interact for. After applying MCTS to protein complexes where we have assumed knowledge of interactions, we now turn to the more challenging and realistic problem of predicting the complexes without knowing interactions, full approach. In addition to the problem of possibly incorrectly identified interacting pairs, this also increases the number of possible erroneous paths dramatically. Anyhow, we find that 91, 175, 52% of structures can be assembled with a median TM score of 0.51, figure 3a, using all possible trimeric interactions with fold dock, while for AFM, only 74 complexes are complete with a median TM score of 0.61. When both trimer approaches have complete assemblies with fold dock n equals 53, the median scores are 0.76 and 0.80 for the full and native trimer approaches respectively. For AFM, the corresponding scores are 0.77 and 0.79, 45 complexes. Using native interactions thereby results in slightly higher scores overall, but to a lower fraction of complete assemblies. Models generated by fold dock outperforms AFM overall, mainly because of fewer complete assemblies from AFM, figure 3a, supplementary table 2. We also included a comparison with multi-Uzard 27 and Haddock 28, providing the real chain structures as input. For Haddock, 77 complexes were completed, with a median TM score of 0.29, supplementary figure 2. Notably, no model has TM score greater than or equal to 0.5. Unfortunately, for multi-Uzard, we were unable to complete the docking for any complex in our dataset methods. To analyze the possibility to distinguish when a complex is assembled to completion and has a high TM score greater than or equal to 0.8, n equals 30, we analyze the ROC curve, figure 3b, as a function of the average interface PLDDT, predicted LDDT from AF, the number interface residues, contacts and interactions between chains normalized with the number of chains in each complex, and the average interface PLDDT times the logarithm of the number of interface contacts. The PLDDT log contacts results in the highest AUC value, 0.77. We fit a sigmoidal curve using the PLDDT log contacts and the TM score, creating the MP-DOC score, multiple interface predicted DOC, see method section. When the MP-DOC tends to be high, so does the TM score and completeness of the complex, figure 3c. This suggests that MP-DOC can be used to select when a complex is complete and how accurate it is. Figure 3d shows the TM score distribution of assembled complexes using all possible trimeric subcomponents from fold dock and examples at different TM score thresholds. The type of symmetry of the complexes and the accuracy of the subcomponents strongly impact the outcome, see below. We find that complexes with any type of symmetry can be assembled with high accuracy, displaying the applicability of MCTS for symmetrical complexes, supplementary figure 3. Obtaining complete complexes with very high TM scores, greater than or equal to 0.8, is critical, as large complexes that are not entirely correct are not likely to provide biologically meaningful insights. It is also possible that some complexes with low TM scores are accurate assemblies, but in a different conformation or biological unit than found in the PDB. For example, 5-TRM has octahedral symmetry but is assembled in a dihedral configuration. Aspects affecting the assembly. To answer why some complexes can be assembled with high accuracy and others not, we analyze the kingdom, the number of total chains, the oligomeric type, hetero or homomer, the number of effective sequences, NEF, the subcomponent accuracy for each complex, the type of symmetry and the interface accuracy between predicted and assembled interfaces, figure 4. We performed this analysis for the complexes assembled with predicted native trimers due to the high redundancy of subcomponents in the blind approach. We use the MSAs and resulting complexes from fold dock, as it is impossible to obtain the paired MSAs and features thereof in the AFM pipeline. The feature representation is created and processed as structures are predicted. Figure 4. Analysis of assembly characteristics using native trimers predicted with fold dock. Figure 4. A TM score per kingdom for the complete assemblies, n equals 58. Bacteria is the kingdom with the highest number of complete assemblies, n equals 29, and reports a median TM score of 0.85. Eukaryota, n equals 17. Viruses, n equals 8, and archaea, n equals 4, have median TM scores of 0.75, 0.44, and 0.92, respectively. BTM score versus the number of chains for the complete assemblies, N equals 58. CTM score versus oligomer type, homomer, N equals 38 out of 114, or heteromer N equals 20 out of 61, using complete assemblies. The homomeric complexes have a median TM score of 0.86 and the heteromeric 0.73. DTM score and NEF. Average TM scores are higher for the complexes with over 500 in average NEF value. ETM score and completion for all complexes, N equals 175. The colored points represent the scores within bins of 10%, and the gray line shows the median for each bin. F. Average TM score of subcomponents versus TM score of the whole complex for the complete assemblies, N equals 58. When the subcomponents display high accuracy, so does the assembled complex. Spearmanner equals 0.80. G. Distribution of TM scores and examples of the best assemblies for each symmetry type. The assemblies are colored by chain, and the true complexes are in structural superposition in gray. The structures shown for each symmetry and the corresponding TM scores are 5 OVS, dihedral, 0.99, 2x2V, cyclic, 0.97, 1 DPS, tetrahedral, 0.98, 1 L0L, asymmetric, 0.75, 1 MFR, octahedral, 0.99, and 5 XBB, helical, 0.88. Full-size image. Bacteria is the most abundant kingdom and displays the highest fraction of complete assemblies, 2985, with a median TM score of 0.85, figure 4a. Eukaryota, viruses and archaea have 1763, 8/12, and 415, with median TM scores of 0.75, 0.44, and 0.92, respectively. Most complete assemblies have fewer chains and are of homomeric type, figure 5b-c, although the spread in TM score is large. The TM scores are higher for the complexes with higher, over 500, average NEF values, which corresponds well with findings for heterodimeric complexes 12, figure 4d. When analyzing how far toward completion the assemblies go, one finds that most complexes are 90 to 100% complete, figure 4e. There appears to be a weak decreasing trend in TM score with completion, suggesting that smaller subcomplexes may be accurate, although the complete complex cannot be assembled. The average TM score of the subcomponents, figure 4f, provides the most evident explanation of when an assembled complex is accurate. When the subcomponents display high accuracy, so does the assembled complex. This is true for both complete and incomplete assemblies and highly accurate complexes. TM score greater than or equal to 0.8 can be selected with AUCs of 0.88 and 0.85, respectively, supplementary figure 5. Figure 5, comparison of assembly with MCTS versus AlphaFold multimer on complexes with 4 to 9 chains, figure 5. Swarm plots displaying the TM scores, N equals 278, N equals 50 for each oligomer except for the nonomers, which have N equals 28, for assemblies using all possible tremors predicted with FoldDoc, MCTS, and AFM end-to-end, E2E. Each point represents one complex with the mean TM scores marked by a black X. The points at 0 for MCTS are those complexes that could not be assembled to completion, N equals 62, and those for AFM E2E that were out of memory, N equals 67. The averages are 0.47 versus 0.58, 0.50 versus 0.54, 0.46 versus 0.56, 0.49 versus 0.67, 0.39 versus 0.41, and 0.35 versus 0.46 for AFM E2E versus MCTS 4 to 9 chains, respectively. Full-size image. The symmetry of the complexes is also found to be significant, figure 4 grams. The dominating symmetry, dihedral, is also the one with the highest number of complete assemblies, 2770 complete, median TM score equals 0.80, while very few asymmetric complexes, 226, are assembled to completion. These have low TM scores, median equals 0.49, suggesting that only symmetrical complexes can be assembled successfully using subcomponents and MCTS. Median TM score for all complexes with any symmetry equals 0.80, n equals 58. The asymmetric complex displayed in Figure 4 grams, 1L0L, https://www.rcsb.org/structure/1L0L, has a TM score of 0.75 however, and most chains are in their correct positions with the exception of one membrane helix and a small cytosolic chain causing the decrease in TM score. The only octahedral complete assembly has a TM score of 0.99, and the tetrahedral median is 0.92. The two complexes with helical symmetry display varying quality, TM scores of 0.37 and 0.88 respectively. All types of symmetry display successful examples, Figure 4 grams, suggesting that MCTS can assemble subcomponents successfully as long as some type of symmetry exists in the complex. The TM score distribution divided by symmetry for the all-trimer approach using fold dock follows a similar relationship to the one found here using the guided trimer approach, supplementary Figure 3. As the assembly proceeds, new interfaces are generated. To provide an estimate of the quality of the assembled interfaces, we compare these to those predicted directly with fold dock using the Docker score 29. Only the complexes that could be assembled to completion and that had interfaces resulting from the assembly were analyzed, n equals 55. The predicted interfaces are found to be of higher quality on average, doc equals 0.34 versus 0.24 for predicted and assembled interfaces respectively. Assembling complexes with 4 to 9 chains. So far, we have only addressed the possibility to assemble complexes with 10 to 30 chains outside of current computational limitations. However, it is possible that the assembly approach with MCTS can be used for smaller complexes as well. To analyze the possibility to improve the accuracy of AFM end-to-end, E2E, on smaller complexes, we created a set of 278 complexes with 4 to 9 chains that have less than 30% sequence identity to the proteins in the AFM training set. See methods. We predicted all possible trimeric subcomponents using fold dock and assembled them using MCTS. We find that the performance of AFM E2E is higher than the MCTS assembly across all oligomers of 4 to 9 chains, Figure 5. The performance is quite consistent for 4 to 7 chains, but drops for 8 to 9 chains, averages of 0.47 versus 0.58, 0.50 versus 0.54, 0.46 versus 0.56, 0.49 versus 0.67, 0.39 versus 0.41, and 0.35 versus 0.46 for AFM E2E versus MCTS 4 to 9 chains, respectively. The performance is low on average, suggesting there is much room for improvement and that predicting the structure of even small protein complexes is a problem that is not yet solved. Discussion. To predict the structure of large complexes directly from sequence information is currently a difficult challenge. Here, we present a method that suggests that one possible approach is to predict subcomponents and assemble them into a larger complex. Using subcomponents predicted from native dimers, native trimers, and all possible trimeric interactions, the median TM scores are 0.77, 0.80, and 0.50, respectively, 15, 58, and 91 complete out of 175 complexes. The scoring function MP-doc can distinguish if assemblies are complete and predict their accuracy, making the blind approach with all trimers feasible. Fold dock based on AF outperforms AFM in predicting trimeric subcomponents in combination with being much faster, 2 to 4 times methods. It can also be noted that AF was not trained for assembly of proteins either, yielding support to the robustness of this method. We find that when the subcomponents are accurately predicted using native trimers, so are the complete assemblies. This suggests it is possible to assemble complexes as long as their subcomponents are accurate. The symmetry of the complex affects the outcome, and some symmetries, dihedral and cyclic, are more abundant and easier to predict than others, helical, asymmetric. Not all tremors can be folded using two NVIDIA A100 Tensor Core GPUs with 40 GB of RAM each. The limit of AF and AFM on this computational platform appears to be roughly 3,000 residues, and 175, 42% of all complexes are larger than that. We find that the assembly approach suggested here is outperformed by the current state-of-the-art AFM for complexes with 4 to 9 chains whenever it is possible to run AFM. In summary, we have shown that assembling large complexes with different symmetries is possible using only protein sequence information and stoichiometry. Modeling large complexes in parts and assembling them converts the problem of predicting large complexes to the prediction of their subcomponents. This suggests an exciting future where models of all protein complexes in entire cells may be modeled. One limitation of predicting protein complexes using the approach proposed here is stoichiometry. It is often not known how many copies of a protein are in a given complex, a requirement for the assembly. Once this limitation is overcome either by computational or experimental studies of complexes, it will be possible to assemble many different protein complexes, possibly in novel configurations. Methods: non-redundant complexes with 10 to 30 chains from the PDB. Since AlphaFold multimer has a limit of 9 chains or 1,536 residues 20 in its training and testing data, and there is no available method validated for modeling larger complexes, we obtained all complexes with 10 to 30 chains from the PDB on January 10, 2022, to extend the current limit, Figure 6a. First, we selected all complexes not containing nucleic acids with less than or equal to 3A ring angstrom resolution, and with the experimental method X-ray crystallography or electron microscopy, 1216. From these complexes, we require all chains to originate from the same organism, 1027. We cluster all sequences from the complexes on 20% sequence identity using MSeqs2, version EDB 8223D 1 YA 07385 63 D4F 103 off 0 EB12B 2058E 30, using this command. Figure 6, Data Selection Process and Statistics. Figure 6, a outline of the data selection process. B. Distribution of the number of chains for the 175 complexes. Most complexes have 10 to 12 chains. C. Distribution of the number of interactions between all chains in a complex. N equals 175 complexes. On average, there are 22 interactions per complex. D. Distribution of the number of contacts per interaction. N equals 175 complexes. On average, there are 70 contacts per pair of interacting chains. E. Distribution of the symmetry types of the complexes. N equals 175 complexes. Dihedral complexes are the most common, followed by cyclic and asymmetric. Full-size image. MSeqs, 2 easy cluster fastafile outname / TMP, MinSec id, 0.2C 0.8 of mode 1. Using clustering, we ensure that no complex has all of its clusters overlapping with any other. We keep the complexes with the most clusters, resulting in the removal of subcomponents of larger clusters, 265. For example, if the sequences from complex 1 map to clusters A, B, and C, and those of complex 2 map to clusters A, B, C, and D, then complex 2 will be kept and complex 1 excluded. After the clustering, we ensure that no complex contains any chain shorter than 50 residues, 193 complexes, to remove protein-peptide interactions. We then download the first biological assembly, 31, from each complex and check that the reported stoichiometry is correct and that the PDB files do not contain discontinuous chains, resulting in a total of 175 complexes. The distribution of the number of chains, the number of interactions between chains, and the number of contacts per interaction can be seen in Figure 6bD respectively. Most complexes have 10 to 12 chains, an average of 22 interactions and 70 contacts between each pair of interacting chains. The symmetry definitions were taken from the PDB annotation, Global Symmetry, Figure 6e. Interaction Network. To create interaction networks for the guided assembly of the complexes, interactions between different chains with CBs, Ca for glycine, within eta ring from each other were extracted. Interactions are defined when 10% of the beta carbons, alpha carbon for glycine, of the shortest of two protein chains are within eta ring from the other. On average, each interaction pair consists of 70 residue pairs, and within each complex, there are 22 interacting pairs of chains, Figure 6c, d. Subcomponent and edge complexity. To assemble entire complexes, we predict all dimeric and trimeric interactions in a set of N chains. The number of possible dimers follows. 1. The number of possible trimers follows. 2. From these dimers and trimers, we extract all edges, pairwise interactions. The number of edges in Dn dimers is Den and in Tn trimers. 3. Structural predictions of dimeric and trimeric subcomponents. AlphaFold multimer v2.0, 20 was run using all 5 models, with 1 structure per model, resulting in a total of 5 structures per prediction, where the top-ranked one was selected for subsequent analyses. 4 different MSAs are created by searching various databases with several genetic search programs. Using JACMR from FMR 332, 3 different MSAs are created through searching the databases on IRF 90v.2020_0133, Uniprot v.2021_434, and Magnify v.2018_1235. The 4th MSA is created by searching the Big Fantastic Database 36, BFD, from https://bfd.mseeks.com/ and Uniclus 30_2018_0837, jointly with Hablitz 38, from HHSuite, v.3.0, beta.3 version July 14, 2017. By using the species and genetic positional information, the results from the Uniprot search are paired. All results from the other searches are instead block diagonalized. All of the created MSAs, 1 paired and 3 block diagonalized, are used to predict the structure of a protein complex. The FoldDoc Protocol 12, based on AlphaFold v2.0.11, was run as well. This protocol creates 2 MSAs constructed from a single search with Hablitz 38 version 3.1.0 against Uniclus 30_2018_0837 using the options Hablitz E0.001, all OMN2. The first of the 2 MSAs is constructed by extracting the organism identifiers, OX, from the resulting EEM file and pairing sequences using the top hit from each OX. The second is constructed by block diagonalizing the resulting EEM file. An extension to 3 chains was made here also, following the same pairing and block diagonalizing procedure as has been done for 2 chains. The folding was performed using AlphaFold model _1, 10 recycles, and 1 ensemble structure. The recycles refer to how many times the intermediate output is fed back into the network, and the MSAs are resampled. The ensemble structure entails how many times the information within the network is processed before it is averaged. The structural prediction was performed on 2 NVIDIA A100 Tensor Core GPUs, each with 40 GB of RAM, with a time limit of 24H per prediction. 3 sets of different subcomponents for the complexes with 10 to 30 chains were modeled, all native dimeric, all native trimeric, and all possible trimeric subcomponents. For the complexes with 4 to 9 chains, all possible trimeric subcomponents were modeled to compare with predicting these directly using AFM E2E. Structural prediction limitations. For the complexes with 10 to 30 chains, the unique dimer subcomponents of 656 could be predicted for AFM and FoldDoc respectively. For FoldDoc, 2,242, 2,246 unique native trimers were predicted, and 2,234, 2,246 for AFM. The 4 that did not work using FoldDoc had the error message, "Cannot create a tensor proto whose content is larger than 2 GB." and the 13 that did not work for AFM ran out of time on two NVIDIA A100 Tensor Core GPUs each, with 40 GB of RAM, with a time limit of 24H per prediction. For the approach using all trimmers, 8049 unique subcomponents were successfully modeled using fold dock. The all-trimer approach using AFM resulted in 7,999 8049 unique subcomponents due to the 24-hour time limitation. For the 278 selected complexes of 4 to 9 chains, see section non-redundant complexes from the PDB with 4 to 9 chains without homology to the AFM training set, there were 2,400 subcomponents for the all-trimer approach, of which 2,375 could be predicted with fold dock. Memory limitations resulted in that 25 could not be predicted. In total, 62 complexes could not be assembled to completion using MCTS. For AFM E2E, 67 complexes could not be predicted due to memory limitations. 8 Intel Xeon E5-2690 V4 CPUs, with a total of 82 GB memory for the MSA generation and the same GPU limitations as above. For the analysis of the entire dataset, N equals 1733, supplementary figure 4, of non-redundant complexes with 2 to 9 chains predicted with AFM E2E, 207 failed, 188 failed due to MSA memory limitations, and 19 failed due to GPU memory limitations using the same hardware as above, resulting in 1526 successful oligomers in total. Path complexity. When considering all possible interactions in a complex, both dimeric and trimeric, one quickly realizes that there are many possible paths that could connect all chains. Take the example of the maximum number of chains modeled here, 30. In the most extreme scenario, all of these are assumed to interact with each other. This means that starting at chain 1, it is possible to attach chains 2 to 30, 29 possibilities, and from these 28 possibilities for each node and so on. If there are no overlapping interfaces in a complex of N nodes and and edges, the number of unique paths that contain all nodes follow. 4. Note that N here is the number of nodes extracted from the predicted subcomponents, which are more than the number of unique nodes, since for example the trimmers ABC and ABD both contain the nodes A and B. Equation 4 is exponential and thereby grows very fast. However, the overlaps will grow with the number of nodes as well, as it will be more likely to have overlapping interfaces with more edges. According to equation 2, there are possible trimmers for a complex of 30 chains. For each trimer, there are 3 possible edges, resulting in 4063 equals 12,180 edges in total. This means that the number of effective nodes is higher than the actual number of nodes. This is because, for example, chain A occurs many times in different trimmers. For example, ABC, ABD, AB all have the possibility to have different interactions between A and B. Following equation 4, there will be 3028, 2.31041 possible paths at the upper bound considering all dimers from 30 protein chains, and many more considering all trimmers. This is a very large number that is not possible to search in a feasible amount of time with our available computational resources. However, it is very unlikely this number of paths has to be explored due to overlaps in the subassemblies. When the subpaths that contain overlaps are excluded during assembly, the number of possible paths reduces quickly. Let's assume there are only 3 possible interactions for each chain. Then the number of possible paths becomes much fewer, depending on how the network is connected. If all branches in a network contain unique chains, figures there is in fact only one possible path that connects all chains. Still, there may be many possible paths to traverse to find this non-overlapping one that connects all chains. Therefore, we limit the number of paths searched at a given time point. Figure 7. Branch network of 30 chains all connected to 2 other chains. Figure 7. There is only one path that connects all 30 chains, the network itself. Full-size image. Assembly procedure with Monte Carlo tree search. From the interactions in the predicted subcomponents, we add chains sequentially following a path through the interaction network graph constructed using MCTS-24. MCTS applies a heuristic search method through a graph to find an optimal path, Figure 8. MCTS consists of 4 different steps: selection, expansion, simulation, and backpropagation. It has been shown that sampling random paths to completion from a certain node, simulation, informs the best action at a certain position. To add new chains to a path, we use BioPython's SVD Superimposer 39. As an example, if 2 pairwise interactions are AB and BC, we assemble the complex ABC by superposing chain B from AB and BC and rotating the missing chain C to its correct relative position. The MCTS procedure is outlined accordingly. For pseudocode, see pseudocode for the Monte Carlo tree search algorithm below. 1. Selection. Start at a randomly chosen node 1, for example, chain A. 2. Expansion. Obtain all edges E1, N, deemed children to node 1 and create N different paths. Expand the new nodes added through the edges by randomly selecting new edges. If the new nodes do not have any edges, they are deemed leaf nodes. In this case, the best scoring node according to equation 5 is selected, and a new expansion is started from there. We expand all possibilities, ensuring convergence towards the best node selection at each position. 3. Simulation. Add chains randomly to the path until the overlap criterion is obtained or the complex is complete. An overlap is defined as when over 50% of the alpha carbons in the shortest of 2 protein chains are within 5 ring from each other. 4. Backpropagation. Score the simulated complex using equation 6. Update all parent nodes with this score. The simulation and backpropagation together provide an estimate of how well the parent node performs in terms of creating a successful assembly path. Figure 8. Monte Carlo tree search, MCTS, procedure. Figure 8. Starting at node A, a connecting node, chain, is selected and added according to its predicted orientation. If this node is a leaf node, a node that has not been expanded before, an expansion is performed. During the expansion, a new node is added, and from this, an entire complex is simulated. The score from the simulation, equation 6, is backpropagated to all parent nodes of the expansion, which is used to determine the UCB, equation 5, and thus select the best possible path. Full-size image. The best child nodes are selected using the upper confidence bound, UCB, accordingly. Phi where Vi is the average complex score, equation 6, of all nodes below node I, N is the number of times the parent node has been visited, and Ni is the number of times the node being scored has been visited. The MCTS procedure is continued until all chains are complex, or there are no more non-overlapping chains to add to the current path, after which the procedure is terminated. The complex 6LNI could not be assembled using trimmers due to no interactions between the chains being present in the predictions. This protein is an amyloid protein, and should thereby not occur naturally in the cell. For the assembly approach using all possible trimmers, there are very many paths to assemble for some complexes. 6 additional complexes, 1 MFR, 1 S3Q, 3 OJ5, 6 J0B, 6 NHT, and 6 PYD could not be assembled due to time constraints, 72H on a 2.6 GHz processor. Pseudocode for the Monte Carlo tree search algorithm. Input. Predicted edges and their structures. Output. Largest possible assembled complex. 1. Select an edge connected to node, chain A, and make this the current node N. While edge underscore available N. Hashtag. Expand. While not is underscore leaf in N equals best underscore out in end. Hashtag. Simulate and score using UCB score equals roll underscore out in hashtag. Back propagate the score. Back underscore propagate score end. Hashtag. Return the path taken to reach the current node N. Return get underscore N, scoring. We score the interfaces of the complexes being assembled in the MCTS using, as done when calculating the PDoc score 12. This score for multiple interfaces we deem Multiple Interface Predicted Doc, or MPDoc. The interface contacts are taken as beta carbons, alpha carbons for glycine, from two different protein chains being within a ring of each other. These metrics are calculated for the entire interface of each chain, as in the Doc29 score for multiple interfaces. For example, if chain A interacts with both chains B and C, the score is taken over both of these interfaces simultaneously. This is done for all interfaces and chains and summed over the entire complex. The complexes with the highest sums are favored. Favoring complexes with higher scores results in complexes with both larger interfaces and with more reliably predicted residues. Sigmoidal fit for MPDoc. To create a continuous score for the multiple interface doc, MPDoc, we fit a simple sigmoidal function towards the TM score, figure 3c, using the complete complexes assembled from trimeric subcomponents and curve underscore fit from SciPy v.1.